Let's learn the material of this magical little creature virus. The word virus comes from Latin virion, which means poison. The discovery of the virus evolved over time, from 1883 to 1935. The characteristics of the virus: can multiply in living cells, have one nucleic acid, DNA or RNA only, are like inanimate objects, very small in size. Virus form: stem, bullet, round, thread, polyhedral, and like the letter T. The structure of the virus: head, capsid, nucleic acid, neck, tail. History of virus discovery. The virus originates from Latin virion, which means poison. Then, how did the virus begin? 1. Adolf Meyer. The history of the discovery of the virus began in 1883, after a German scientist, Adolf Meyer, discovered the existence of yellow spots on tobacco leaves. Knowing that, Meyer tried extracting the tobacco sap and then spraying it on tobacco that was still healthy. Apparently, healthy tobacco also experiences yellow spots. Then, Meyer examined the tobacco sap using a microscope, apparently found no bacteria, so he concluded that the creatures that attack tobacco are smaller than bacteria. 2. Dmitry Ivanovsky. In 1892, Russian scientist Dmitry Ivanovsky carried out the same study as Meyer, which filters sick tobacco sap. The difference: Dmitry filters the sap with a bacterial filter. Then the filter results are sprayed on healthy tobacco. It turns out tobacco also becomes sick. 3. Martinus Beijerinck. Beijerinck is a Dutch scientist who did the same research with two previous researchers. Except that Beijerinck tried to deactivate the creatures that cause the disease using alcohol. The result is alcohol cannot deactivate the creature. Beijerinck called it a virus escape filter. 4. Wendell Meredith Stanley. A scientist from the United States, Stanley successfully crystallized creatures that cause disease in tobacco in 1935. Then, the disease was named tobacco mosaic virus (TMV). Characteristics of viruses. The characteristics of the virus are as follows. 1. Viruses can be like living things, for example, they can multiply if they are inside living cells. 2. Having only one nucleic acid, DNA or RNA. 3. Viruses can be like inanimate objects, for example, not doing metabolism, not breathing, not moving. And in the form of crystals if they are outside living cells. 4. Very small in size, which is between 20 and 300 nanometers. Virus form. Apparently, viruses take a variety of forms, you know. Want to know anything? 1. Rod-shaped, for example TMV, tobacco mosaic virus. 2. Rod-shaped and oval-shaped like a bullet, for example rhabdovirus. 3. Round. For example HIV, human immunodeficiency virus, and orthomyxovirus. 4. Shaped laminar thread, for example the Ebola virus. 5. Shaped polyhedral, for example adenovirus. 6. Shaped like the letter T. For example bacteriophages, which are viruses that attack the Escherichia coli bacteria. Here is the picture. Virus structure. Viruses are not classified in cellular organisms because they do not have cell parts, such as cell walls, cell membranes, cytoplasm, and other cell organelles. The bacteriophageal body structure is as follows. 1. Chief. The inner head contains nucleic acids, while the outer parts are covered by capsid. For bacteriophage viruses, the head is polyhedral with a type of nucleic acid DNA. 2. Capsules. The capsid is an outer sheath of the virus which contains many protein subunits called capsomeres. Capsid consists of several forms, so it influences the shape of the virus. 3. Nucleic acid. There are only one nucleic acid in the virus, namely DNA or RNA. It is these nucleic acids which later serve as genetic information for replication. 4. Neck. The neck is the link between the head and tail. The neck functions as an outlet for nucleic acids leading to the tail. 5. Tails. The tail of the virus consists of tail fibers and base plates. This tail functions to stick to the host. The following are virus structures other than bacteriophages that have been found. How to live a virus. Viruses are classified as obligate intracellular parasites because they can only live inside living cells. That is, if the cell dies, the virus will not die but crystallizes. Live cells that carry viruses are called host cells. How does the virus recognize its host? Namely using a lock-key system or suitability. Based on the type, host cells are divided into two: broad host range and narrow host range. Viruses with a wide range of hosts can infect a number of hosts. For example, bird flu viruses can infect birds, pigs, and humans. Whereas viruses with a narrow range of hosts can only infect certain hosts. For example, the U virus only infects cells in the respiratory tract, and bacteriophage viruses can only infect Escherichia coli bacteria. Transmission of the virus from one host to another can be through air, mucus, water, blood, or through intermediaries such as mosquitoes. Propagation of viruses. The proliferation of viruses is known as self-replication. For viruses, host cells are a source of energy for protein synthesis. Viral propagation is divided into two, namely lytic and lysogenic cycles. 1. Lytic cycle. Lytic cycle is caused by the resistance of host cells is weaker than the power of viral infection. As a result, the host cell will burst and die, and will produce new virions. The stages in the lytic cycle are adsorption, penetration, synthesis and replication, maturation or assembly, and lysis. 2. Lysogenic cycle. Lysogenic cycles occur if the host's body's defenses are stronger than the virus's infection power. In this cycle, the host cell can still reproduce normally and will not break immediately. However, bacteriophageal viral DNA will interact with the chromosomes of the host cell to form the profile. When the host cell containing the prophag divides, then the prophag will be passed on to the next cell. The stages in the lysogenic cycle are adsorption and infection, penetration, fusion, division. Synthesis. To understand more, please see the following picture. Quiperian. Virus classification. The classification of viruses is quite a lot, amounting to 6. Everything is based on the similarity of features possessed. Want to know more? 1. Virus classification based on the presence or absence of a nucleocapsid. There are two groups of viruses in this classification, which are as follows. The sheathed virus is a virus which consists of lipoprotein and glycoprotein, for example poxyvirus, herpesvirus, togavirus, rhabdovirus, and paramyxovirus. Naked virus is a virus that has no envelope on its nucleocapsids, for example papovirus, adenovirus, picornavirus, and reovirus. 2. Classification of viruses based on the number of capsomers. There are 5 groups of viruses in this classification, which are as follows. 1. Viruses with 32 capsomers, for example parvovirus. 2. Viruses with 60 capsomers, for example picornavirus. 3. Viruses with 72 capsomers, for example papovirus. 4. Viruses with 162 capsomers, for example herpes virus.